This is a production of Cornell University.
Great, thank you. Thank you, Colin. It's a great pleasure to be here to share. Well, I feel actually I'm coming home actually because I used to be section of horticulture, right? So it's a really great pleasure to share some of our recent researches on sort of broccoli. Not all you can see actually. I just share part of our research on broccoli. And the title you can see actually molecular mechanisms of plant response to warmer temperature. So I was sort of struggling whether to use. Let me see whether I could have my pointer. I tried to use a pointer. The warmer actually or warming temperature. So it's I have been it works.
I think so.
Okay, that's great. Thank you.
In theory.
Yeah. Okay. So I have been debating actually whether use warming. At the very beginning, I gave Karen actually the warming temperature. Then I changed to 5. Last minute I changed to warmer temperature. But essentially saying actually, speaking of warming temperature, we immediately realize, why doesn't the global warming? Okay, so you will see actually global warming. You say, well, global warming generally refers to certain maybe 50 years or whatever actually period the global temperature increase. So you can see here, it's barely 1 or 2 degrees difference. So what matters, right? Especially this is sort of introductory slides. You will see actually this is global warming sort of diagram and a little bit exaggerated perhaps. But that being said, you will see actually this is warming really matters. Here is actually 1 week ago, actually just published when there will be strong stratospheric warming actually events happens, then will cause actually polar vortex. Then polar vortex actually, serious sort of tropospheric temperature will be dramatically changed actually. Certain area will be surely much warmer than it used to be, and of course sometimes certain area will be cooler than it used to be. Okay, so the warming, the temperature fluctuation is real, it's dramatic. So warming really matters, as a matter of fact, not simply just 1 or 2 degree differences. So the one of story is polar bear, because of the global warming and force the polar bear migrate to land area. And as you can see here, and then actually where the land actually where brown bear actually habitates. And then when Harry met Sally, what's happened, you guys know, that is actually have baby here, okay, called a polar bear, okay. Now this is actually last November 17 news, actually. It's recent news. They use this as an example, actually, to show global warming. But I use a separate, sort of in reverse sort of direction. And that is why the warming may be a good thing for this new baby, right? So this is, but warming generally do more harm than beneficial. Okay, this is December 6 Cornell Chronicle news. So warming will lead to that sort of New York, upstate New York sort of lakes oxygen content such as dramatically reduced. Okay. Now this is oxygen in lake, but in terms of the warming on plants, so the warming effect on plants could accelerate plants develop growth and development, or actually delay the growth and development. Okay. And in terms of the effect on vegetative growth, it will generally warming will promote cell elongation, and which will lead to the weaker stems or petioles. Okay, so weaker plants you can see here. But the warming temperature will have more profound impact on reproductive growth. As you can see here, actually, it will promote earlier flowering. The transition to flower could be earlier, and significantly reduced pollen production, and also actually pollen germination was significantly inhibit pollen germination and fertilization, thus actually significantly reduce the seed yield. Okay, so now in terms of today, I said use broccoli as example. You know, Thomas, I know Thomas is online. Thomas Buchman actually did the work actually years ago, and the high temperature, okay, on the broccoli. So we're talking about broccoli today. You can see in greenhouse or in field, in greenhouse. So both of high temperature, you know, this is field about 30 degrees Celsius, and in greenhouse about 35 degrees Celsius, you can see here. But will cause actually uneven appearance of head, you know, broccoli head, or actually severely injured head had predominantly small buds, you know, sort of quantity and the quality will be significantly impact, okay, high temperature. And another example, that was Galaxy actually, broccoli, but in terms of green harmony broccoli, you can see here, January 16 degree, produce actually normal broccoli head we are consuming. And then if we grow at 28 degrees Celsius, you see the produce actually cauliflower-like code, okay? While grow at 22 degrees, you will see produce intermediate head, okay, code, we call it code. Okay, now this was work was done actually by also Thomas Buchman's lab, actually with graduate student, former graduate student Denise de Cruz, actually, who actually used to work in my lab, as a matter of fact, to dissect the molecular mechanisms underlying this sort of change. So what Denise did actually that time, actually, I think she spent half a year or 1 year in my lab actually to provide sort of those flower identity genes during the broccoli head development. So try to see what's going on with related to that. And to be honest with you, that time I was very, very critical regarding this work. I thought that this work is just wouldn't really reveal the underlying molecular mechanisms. You just you test actually what gene here. Of course, this gene compared with this way, gene expressed here, of course, wouldn't express there, right? So you provide, it really didn't mean too much. So I was very critical, and Denise was really unhappy actually about this. So I'm sorry actually to Denise, but that was the fact, okay? So, and I gave her a hard time actually during defense because I was sitting on her thesis defense too. And Denise, you know, as I said, actually provide use PCR analyze different gene expression at the different stages of those actually sort of broccoli head or abnormal broccoli head. You can imagine, as you can see, actually certain gene not expressed here, of course not expressed there, right? So it's really not so insightful, okay? No, this is a really good actually review, illustration of that about the flower development and the regulatory genes involved in this one. Yes, you can see here, different genes, pillar2, you know, those sort of A gene, ABC gene, the model you guys perhaps know actually, right? Then, and what's happened actually, you see actually this stage is sort of inflorescence meristem, actually, this cauliflower, so-called IM, okay? This flower development at this stage is inflorescence meristem at 16 degrees. That's equivalent to that broccoli development. You can see here, okay? So IM. Later, we will frequently use this term. And this stage is intermediate stage, equivalent to actually 22 degree. Okay, that's for all primordial stage. Okay, that's what the nature of that intermediate code. And fluorobads code called the FB stage. As you can see, that's what the broccoli we are consuming, right? That's what on shelf on the grocery store you will see. Actually, that's FB stage. So you will see IM stage, FB stage, and the FP stage. So different temperature equivalent to this development. As I said, actually, why I was so critical, because let's say if you're going to profiling the expression of AP3 gene, of course, you know, if we stop here, there's no expression there at all. Okay, so you sample the sample and it says, oh, there's no AP expression here, but expression here. So what's that mean? Of course, they shouldn't have any expression, right? So that's why I was said not so insightful or informative actually. So that's why what I mean actually, I was so critical. Okay, so but again, actually, this is the terminology you could see 16 degree arrested here. So in other words, you can see very clearly 16 degree IM stage, that's cauliflower meristem, actually, like inflorescence meristem, that's called cauliflower-like code. Okay, so IM stage, then intermediate is really flower development stopped at the FP stage. Okay, then that's what we are talking about actually, what we are eating actually at the FB stage. Now, back to this one actually, I frequently have been asked to say, hey, when media interviewed me actually says, hey, why you get into this project? So that was related to actually Professor actually Li Ping Chen from Zhejiang University, and she visited my lab as a visiting scholar in early 2010s actually, and then she returned to Zhejiang University, and 2017 I was invited to visit Zhejiang University. Then we discussed about potential collaborations. Okay, so one of the collaboration we initiated is this work, demethylation, DNA demethylation in leaf senescence, because as I said, actually, Dr. Li Ping Chen is an expert actually on cell grafting and its associated epigenetic sort of mechanism, especially DNA methylations. Okay, so I thought, okay, I work on senescence. You are good at sort of methylation, and let's work together about this gene. So this was published sort of in Molecular Plants years back. And then another actually collaboration work was sort of today's topic. That's mechanisms underlying temperature-induced reproductive growth in broccoli. And so I thought, this is really interesting phenotype, but unfortunately, molecular mechanism is not understood. So I thought, of course, we have another project I didn't list here. That's massive horizontal sort of gene transfer. And that's a very exciting story, actually. We are drafting the manuscript. But this is back to this one. So we are working on this sort of to this project. That's how I get into this project. Okay. Now back to this slides I just showed you. And in order to investigate into this one, so we decided to add an additional stage. That's the transition stage. Okay. From vegetative growth to reproductive growth. So you will see VR called vegetative to reproductive growth. And hereafter, you will frequently see the terminology VR, IM, FP, and FB. Okay, so don't get confused. I'm not going to spell out later, but this is a different stage of flower development. Although this is from Arabidopsis, but it's pretty much the same as in broccoli. Okay. So remember this actually, this terminology. I will not repeat this terminology. So in order to do that, as I said, So this is, well, first we need to repeat whether this phenotype actually shows up. You can see this is surely, okay, 20. So this is a VR stage, okay, 16 degree, 22 degree, 28 degree. You see, this is 28 degree will be arrested or stopped for development stopped or at the IM stage, inflorescence meristem stage, okay? So regardless how many days you just grow, you just stop at this stage, produce cauliflower-like code, okay? Then actually at the 22 degree, you know, that's intermediate. Then 16 degree, of course, that's the broccoli head, you see, okay? Now, at that time, actually, I thought, well, why is it collaboration with her actually about use sort of epigenetic mechanism involved, the DNA methylation? Because I thought at that time, I told her, I said, my gut feeling, sometimes gut feeling is not necessarily true, but in this case, I better right. I told her, I said, epigenetic mechanisms involved, either sort of chromatin modification, that's acetylation, or actually DNA methylation. Okay, so actually I even lined up with one of my former graduate students, now actually a professor at the Peking University, who work on chromatin actually modification, acetylation of those proteins. So, and then another mechanism, I said it's methylation, most likely involving mechanism. So we test the mechanism and use DNA methylation inhibitor, okay? DNA inhibition inhibitor actually, now this is water, we treat with water, you can see 24 days, 31 days, so all remains 28 degree. This is a 28 degree, remains as cauliflower-like cold, okay? That's same. However, if we exactly say at 28 degree, no, that's 28 degree, right? It's just stopped at this inflorescence meristem. But if we treat with DNA methylation inhibitor, you will see actually this is developed sort of intermediate and then broccoli head, right? So in other words, even at 28 degree, we use DNA methylation inhibitor. So this actually flower developed as if were grown at 16 degree, right? So that means, no, before I get your conclusion, so it's about the DNA methylation. DNA methylation generally takes place actually on the cytosine C, then methylated here, as you can see here, right, in the DNA. Okay, so the methylated, and so under the enzymatic sort of methylation, methylates actually. At a very later stage of the mechanism, actually, we need to study later. I will get back to this mechanism. And of course, mechanism, there were two DNA methylation patterns in the G. One is the CG pattern, okay, methylated C, and CHG will be methylated. You know, H stands for could be anything, ATC, okay? So the H, or in the context of CHH. So that will be, you know, C methylated, but HH could be any of those, all right? So I will refer to this a sort of methylation pattern in the context, when I said in context of CG or in context of CHG or CHH. So you will just, you know, part of sort of C that will be methylated, okay? Now that 5 DNA methylation inhibitor is N here. So N, of course, here, this is carbon here, right? This is nitrogen. Of course, there's no way this could be methylated to other methyl groups, right? So that's the mechanism of DNA methylation inhibitors. Hop back to this one. So you can see here, we already showed actually, I already showed that DNA methylation inhibitor just eliminates the temperature-induced actually for cessation or arrest, right? Develop normally. So the conclusion is very, very clear. That is, DNA methylation is involved in the warming temperature-induced change of broccoli head to cauliflower-like coat, right? So you guys have any problem with this conclusion? Okay, so once we know that DNA methylation is involved, then, again, actually my gut feeling was right. And so then the next, of course, is to test, to investigate the global sort of whole genome biosulfate sequence method to measure the whole genome's DNA methylation profiles. Okay, so now this mechanism is called WGBS, called a biosulfate sequence. It's really actually biosulfate will convert C to U, then U will become C when sequenced. Okay, but if methylated, C will remain as C. So by sequence this one, so then you will be able to know which one is methylated, which one is not methylated. Okay, that's the principle of this methylation. So in order to do that, so with this is I showed you. So we decide to harvest sampling. Okay, what sampling stage we decide to harvest is this is certain stage actually we harvest the samples. 28-degree at iron stage, and because there's no way you can harvest, it's the same thing, actually meaningless, right? So there will be actually 2 samples for DNA methylation, global, biosulfate methylation purpose. So the 2 replicates, not 3 replicates, 2 because it's so expensive. And then at the same time, I will get back to this one, where at the same time, we harvest the 3 biological replicates. That means we'll actually have 3 transcriptomes. So in other words, for each point, we have 2 methylones and 3 transcriptomes, okay, for each stage. At the same sample, same stage, we harvest. Now, get back to actually continue to discuss about the methylone. So methylone, all those data we are not going to go over, but you will see actually global dynamic changes. You will see at 28-degree, you will see methylation in the context of CG context. I said the CG context, CHG, and CHH context. That's what previously I told you that sort of, you will see any of them, you see the methylation level is higher than lower temperature, right? So any sort of that's global change. Actually, we're not going to go over. But what I want to draw your attention to is the gene promoter region. So the certain genes and the promoter region and the 3' actually untranslated region. So the promoter region you can see here. Huh? Does not work anymore. Huh?
To turn it on.
Oh, it's here. Okay. So you can see actually this is 28-degree. Okay, the high temperature, the methylation level higher than 22-degree, then actually 16-degree. That's normal produced. So the methylation level change is actually really matters. Now this is a CH context. Okay, this CHG context, same thing actually. Although the CHH context really doesn't matter too much because I didn't see significant increase. Okay, the promoter region, you see the promoter region really methylation level higher than actually. And so of course there's a different stage of this methylone. We're already established that what you need to do, of course, bioinformatics, just a different comparison. Okay, in order to compare, you can imagine we performed all various actually sort of analysis, VR stage, whatever stage to see the methylation pattern change, so on and so forth, 22-degree, 16-degree. Okay, so because of the time, I'm not going to go over, but to simplify today's presentation, I will draw your attention mainly actually focus on the analysis comparison of this transcriptome, this and here, and this comparison of this one in order to get genes related to this, arrested at this stage for genes required for IM So we'll analyze actually this group of analyses. Okay, so to simplify this part. Now, I already said that, methylation changes region. So there's a terminology called differentially methylated region called DMR. Okay, so DMR. What's a DMR? Well, sometimes we use the hypomethylation. For example, normal cell, cancer cell. For you can see here, this is cancer cell. In this cancer cell, this is cancer cell. This DNA is highly methylated compared with normal. Then we call this region is highly methylated. Vice versa, this is sort of hypomethylated. And of course, it could be both high, both low. Those both high, both low, of course, that's irrelevant, right? So we're not going to pay attention to those genes. But just mentioning the methylation changes, okay? So first of all, we need to identify those differential methylated regions, okay? It's called DMRs. And so compare 28-degree versus 16-degree, we have VR stage, you see actually 271 sort of in CG context, okay, CG context. You see actually differentially methylated regions. And, we're not going to go over, the status, but you can see there's various status methylation, certain region change actually up hypomethylated or hypomethylated, okay? And hypomethylated, so different comparison, I already mentioned to you. Told you. So based on this, actually, we have sort of at the same time, we already identified those differentiated method regions, certain numbers of those one. So I will leave this one, move to another actually topic, related topic. That's actually, as I already mentioned, we also have 3 transcriptomes for each sort of stage, right? Each sample stage, we have 3 transcriptomes. So at the same, we'll analyze what genes are expressed, differentially expressed, dramatically changed at a different stage. Okay, you may wonder, okay, then what's the difference actually with the previous, you mentioned the Denise's analysis. Of course, Denise's analysis is sensing just the several genes. This is a whole genome analysis, but this analysis per se wouldn't reveal really molecular mechanisms. Okay, so I will get back to this later talk. So we find that this is called another actually terminology similar to differentially methylated region, DMR. There's another terminology called differentially expressed gene, DEGs. Okay, so expressed in this stage, but not in that stage, vice versa, downregulated, upregulated. Okay, so this comparison, you can see certain message RNAs expressed in this stage, but not in this stage, vice versa. Okay, so you will be able to find those genes dramatically, expression profile changes. So you can see here, this is VR stage, upregulated gene, and downregulated gene. How many upregulated, how many downregulated at different stages, 28-degree versus 16-degree. Various comparison, you will see the different differentially expressed gene profiles. Okay, so you can see actually dramatic, those huge number, 9,000. This is a really huge number. Okay, we did find those numbers, actually huge number of genes. So what matters? Why you do this when it's really difficult to study actually? How many genes? How do you know which gene is responsible for that phenotype I showed previous? Okay, so so we further actually sort of analyze. First of all, we need to get rid of some of the genes that are most unlikely involved in those functions, right? So first of all, compare with this one. So you will see actually only, where we need to consider only 1,600 genes about this one. So at least we could get rid of 600 genes, get rid of this. We don't have to consider about those genes, okay? And similarly, this is downregulated gene, sort of 2,000, nearly 2,000, we get rid of 500, more than 500 genes, and so on and so forth. So we get rid of 1,000, nearly 1,000 and more than 1,000 genes. So only need to consider small number of genes. Okay, so now the small amount of genes we already got to this one. Similarly, as for that previous slides was 28 degree, but for this primordial to fluorobats actually develop genes, could be this kind of sets of genes. That's red, that's blue color actually. So it's hard to interpret why we consider that after the seminar because of time. I could, if you want, I could explain why we focus on those genes. Okay, so back to this one. Now you still have with association of where previously we have DMR, so differentially methylated region gene, right? And we have this DEG, differentially expressed genes. Now, what are those genes that also contains DMR region, so methylated? Because we know methylation matters, okay? So only those genes, differentially methylated gene with differentially methylated region, expressed genes with differentially methylated region, most likely that matters, okay? So we'll work on this one. Now, but you can see here, still there's sort of 318 genes and hypomethylated 18, and it's very similarly to this gene. So still, now the immediate question is, for this stage, where we have so many genes, 318, 200, so still about more than 500. It's very difficult to study, actually. Now, for 22 degree, we have about, nearly 400 genes, hypermethylated and hypomethylated. Now, the immediate question is, hypermethylated matters or hypomethylated matters? That's the first question, okay? Now, the methylation, of course, where I just recall, I showed you the data. DNA methylation inhibitor really actually eliminate those. DNA inhibitor, methylation inhibitor, of course, inhibit DNA methylation. That means if no DNA methylation, that means it's supposed to be highly hypermethylated, right? So then, therefore, actually, the answer to the previous slides is that it's hyper DMR matters, okay? Not the hypo DMR matters. So, but hypomethylated DNA matters. Now, another question is, so therefore it matters, right? So another question is whether upregulated gene matters or downregulated gene matters, right? Because immediately you will ask, well, for this IM stage, required for this is upregulated or downregulated gene matters. Now, how DNA methylation affects gene expression? So generally speaking, oh, geez. I should have slides here. Why didn't it show up? Huh. There you go. Okay, here you go. So you see, actually, generally speaking, that the DNA methylation affects gene expression. How does that? It's a transcription factor we know actually binding to the promoter region and activate gene expression. That's general sort of rule. And however, if the promoter region, this DNA methylation here that prevent transcription factor from binding to this cis element, then of course there's no gene transcription. Okay, so in other words, the DNA methylation inhibits actually suppress gene expression. Okay, so therefore actually another example actually downregulate gene really matters. Another example is that perhaps you guys already know. Oh, why this one? Sure, again. This is Dr. Jin Jivano's publication, I believe. It's in Nature Biotechnology. And you can see here, there's actually RAIN binding proteins. RAIN binding protein binding to these regions here, DNA methylation. And in the wild type, DNA methylation is less. So then actually RAIN transcription factor is a gamma's transcription factor. Okay, we're binding to that and promote actually ripening. But if you have higher methylation, then actually this transcription factor ring couldn't bind to it. Of course, delay, slow down the ripening process. So that really DNA methylation really inhibits or suppresses gene expression. Okay, so again, downregulate DEG matters. Okay, so back to this one. Okay, downregulate gene matters instead of this upregulate. Similarly to here, okay, downregulate gene matters. So in other words, we already narrowed down now to 232 genes that potentially be responsible or required in order for IM develop into FB stage or actual FP stage. Okay, so on and so forth. And 168 genes that is required in order for FB continue to develop into FB stage. Okay, makes sense? So now it's much easier now. So narrow down 200 and 180. So nearly to around 200 genes actually. So, and so that's what I say actually. So, okay. So therefore, so we named this gene called the floral development cessation gene, FCG, okay? Required either for IM to transition to from IN to FP, or that's 28 degree, or FP to FB state, transition those genes, okay, respectively, you can see here. All right, what are those genes you will wonder actually? So this gene, no, this is different gene, of course, the biological processes, cellular components, and the molecular functions, those all gene go, you will see those kind of analyzed. So, but I would like to draw your attention is to this first line, that's ribosome biogenesis related gene. So, okay, this is gene is very, very important, I personally believe, okay? So now this genes, actually, what are those genes? You can see ribosome biogenesis related genes is a different gene. We are not going to go over, but it's very, very important. I will explain why we're focused on those. These genes, ribosome bio, sounded very boring, but it's important as a matter of fact. And similarly, for 22 degree, actually, that's a FCG, is also actually so ribosome biogenesis related genes, okay? So both of those actually you can see very, very predominantly, sort of express really matters, you can see here, okay? So now this is sort of ribosomal biogenesis, why we care about the ribosomal biogenesis related genes. And first of all, actually, these genes are extremely highly expressed actually in I should emphasize, this is the counterpart gene of this broccoli gene, okay, in Arabidopsis, expressed highly in the apex. Should apex, of course, yeah, okay. So I will move really fast now. Okay, so this is really matters, ribosomal biogenesis really matters. You can see here in cancer biology, it's very, very important. It's a hot area in cancer cancer therapies, as a matter of fact, related to cell division, whatever. You can imagine, of course, there's fluorobaths development related to cell division as well, actually, okay? And so with, in plants, you can see here the temperature, there's a sort of review articles. So ribosomal ion related to temperature stress change and the cell cycle arrest, and also developmental abnormalities. Okay, so this is really ribosomal RNA. My gut feeling is when the student analyzed the data, I said, well, let's focus on this. So my gut feeling sometimes works. I bet already once correct. I bet this time will be correct too. Okay, so and DNA methylation. So warmer temperature, hypomethylation, then low FCG expression, because no expression of this gene, of course, the flower development will be stopped, ceased, okay? Now, back to this one actually is, so you will see, well, whether this gene methylation really matters, you will see the correlation, temperature methylation and FCG expression during those different stages, okay? For just IN required for IM to FP stage. You can see here 28 degree, 22, and 18. You see the expression. This is methylation, promoter region methylation. Okay, this is expression level, the digital expression level. Now, the lower temperature and the lower methylation, and the higher the expression, vice versa. Okay, those all those genes. I just show you few of none of all correlated very well. Okay, so those data actually can directly. Now, those genes actually are predicted to be a cis element DNA, a transcription factor binding to this. Okay, this is already this data bioinformatics. You will see what kind of genes, transcription of DNA methylation here, the transcription factor binding to this one. So different transcription factor is are predicted to binding this promoter, especially that the DOF gene actually domain with one finger. That's transcription factor. It's very predominant. You can see. Yeah. Similarly, that makes sense, right? Similarly, for 22 degree, it's the same thing actually. So predicted all those transcription factor binding. So that makes a lot of sense for this one. Now you will wonder actually whether methylation really matters with that expression. Yes, actually we did the DNA inhibitor, methylation inhibitor treatment. You will see actually once you treat with methylation, the DNA methylation is inhibited, then this gene expression is released, the suppression is released. So all those, no exception, okay? So I just show you for 22 degree, similarly, okay? So this is no exception. So that's make a lot of sense actually for this. Okay, so DNA methylation, inhibitor really matters actually for those. Now to summarize, so we just talk about it, 16 degree, grow at 16 degree, then produce cauliflower-like sort of code, and then continue to develop FP, then FB. That's flower buds actually we are consuming. And in order for this IM developing into FP, there's a set of gene called FCG is required, okay, for this gene to develop into this one. And in order for FP to continue to develop to FP, then there's another set of FCG I already mentioned, okay, to required for this develop transition for this one. However, at the 28 degree, causing induced DNA methylation, especially in the promoter region of FCG, to suppress this gene expression. This gene is required, as I said, in order to develop from IM to FP. But this gene, due to the suppression, due to the DNA methylation, then of course this will be stopped here at the IM stage, right? Continue always at this stage. So similarly, so this 22 degree will be very similar to this one, inhibitor to this one, okay? However, if we use DNA methylation inhibitor, then inhibit this methylation, even at the 28 degree, it doesn't matter. They can continue always produce that way, okay? So future directions, I just mentioned, first question is methylation mechanisms. Okay, what are the mechanisms to this? Because this is important. In future, once we identify the DNA, a gene that controls this one, we could manipulate, knock out this gene, doesn't matter. Even at a high temperature, they will not methylate the whole genome. Then actually can grow, yeah, okay? So, and the second question is what are the FCG functions? We could use genome editing to do that. And acknowledgement, and this is did all the work and contributed equally. And Jing Giovanni, actually, I'm really grateful to him. Actually, one night, read my manuscript and provide the insight for actually. And also just for actually FCG name, actually just offered me, actually says, well, why not you call FCG? Okay, so that's a very appreciated useful information. And there's actually dollars. And thanks for the media and different worldwide different medias. And I just try to advertise for molecular horticulture. So we publish really high quality papers in molecular horticulture with immediate impact factor of 5.9, okay? And okay, so I advertise for Ignite failures Cornell have, okay? So graduate student postdocs, Cornell will offer Ignite. I have a startup for the Resilient Incorporation. We work on a lot of things that are in future, I hope we'll be able to share with you guys. Okay, very excited. And that's it, I think. Oh, yeah, that's it. Any questions? I will be happy to answer any question.
A couple of quick questions, maybe?
Yeah.
Jen, can you give us an idea of how long at these different temperatures it takes to start getting the, the change in the molecular upregulation? I mean, so you were running at constant temperatures, right? But under climate change, which is your premise here, what's going to happen under climate change, one hot day, it's more and more likely to happen, especially in where we're growing a lot of these crops. So how long does it take till we start seeing a phenotypic change?
Yeah, that's a good question. Actually, duration, we haven't performed such kind of experiment yet. Yeah, that's a good question though. But a persistent a quarter of, sort of a high temperature is not unusual to see actually nowadays, right? Yeah, any other questions?
Do we have any questions from Geneva?
No?
Yeah, so I see Zach's got his hand up. I can't figure out how to get my hand up, but this is Thomas. So very nice work. So 25 years ago, there were too few genes. We only had like 5, and then quickly went to 5,000, which made it impossible to really investigate from the other side. So it's beautiful how you've brought that down to a tractable number. And that really has facilitated progress on this question, and being able to do the global analysis from just being too big and saying it's too much to deal with. So that is nice. So
you say that Zach has, thank you though, actually. Zach, you say, Zach has a question, you say?
Yeah, so back in the day, it was all about the expression of the gene called cauliflower. And I wonder if you took an extra look at that particular gene to see whether it was playing a role in this. To go back to the—
That's a very good question, actually. I mean to say that, actually, because you can predict the effect based on a model. Those genes, a set of sort of FCG genes will be really suppressed in the cauliflower plants.
Right.
So it's easy to test, as a matter of fact.
Yeah, and we keep coming back to it. Probably only matters occasionally and not in this particular genetic system. But the story keeps recurring, so you have to come back. It's such a beautiful, simple story. Just doesn't happen to be a correct story.
Yeah,
so
thank you, Thomas. And but I personally believe actually this mechanism is conserved, as a matter of fact, for other crops as well. Yeah.
No, exactly. I mean, this is such a great system to study it in, because it really separates things out in time developmentally, and the tissues are relatively large. So horticulture gives good model systems. That's a good theme.
Do we have any other questions? Yeah, in the back.
Are the methylation blockers something that are or you think could be used in the field to make the crops more resilient?
You mean the inhibitor?
Yeah.
No, actually, because actually this chemical 5-0C actually is used for cancer therapy. Actually, it's toxic, as a matter of fact, to inhibit methylation. I showed you cancer, right? Some hypomethylated. This inhibitor actually used to suppress this sort of suppression, to reverse the suppression. It's a matter part of that cancer chemotherapy, as a matter of fact. Yeah, it's toxic. Yeah.
Okay. Well, let's give.
Yeah, thank you. Thank you. Yeah. Anytime, you guys should have my email address. Could email me or stop by 140 Emerson Hall anytime. Okay. Especially graduate student, if you're interested in Ignite failure very well paid actually, worker interested in entrepreneurship, please contact me, okay? Yeah, thank you.
This has been a production of Cornell University, on the web at cornell.edu.